Hi, everybody. Welcome to my home. I have a different recording location tonight. I was lucky enough to get the kids in bed early and the dogs out back. So hopefully we have a nice, quiet environment that rarely happens in a home that contains 3 toddlers. So we're going to talk today about gene mutation. Mutations in the DNA are changes in the genetic material, and they can happen in just a single gene or in a chunk of the whole chromosome or the whole chromosome. So mutations that occur in just a single location are known as gene mutations or point mutations, and ones that change the whole chromosome are known as chromosomal mutations, and we're going to go through all of the different kinds. A gene mutation involving a change in one or a few nucleotides are known as point mutations because they occur at a single point in the DNA sequence. A lot of times we refer to that as occurring with just one letter or impacting one codon or just a single base change. Point mutations include substitutions, insertions, and deletions. Again, they happen at a single nucleotide point in the DNA. In the case of a substitution, one base changes or substitutes for another. For example, you could substitute out the base adenine for guanine. Now, adenine does not pair to guanine, it pairs to thymine, but a substitution change would be just the base getting replaced or substituted out with a different one that it wasn't originally there. An insertion would be an additional base getting inserted into the strand, and as a result of that being inserted in there, everything else would move over one letter to the right, or however many letters were inserted. A deletion is when at least one base is removed, and then everything would move at least one letter to the left or however many it had removed. So this is an example of a substitution, and the definition here, substitutions usually affect no more than a single amino acid, meaning that they change no more than one codon, and every codon codes for a specific amino acid. So here you have your DNA strand at the very top, TAC-GCA, and you can see that the C is in red, so that's what we're going to change. In mRNA, you have, as a result of that, AUG, CGU, and the amino acids that it produces are methionine and then arginine. When you substitute that out, and so instead having TAC-GTA below, change the C to a T, that then changes the mRNA sequence to AUG-CAU, and it changes the amino acids to methionine and then histidine. So the only amino acids that changed were the arginine to histidine, and that the rest of the strand remains the same. The effect of insertion or deletions are much more dramatic when they produce what is known as a frame shift mutation. They impact every single codon, so they will change every amino acid from the point of the insertion or deletion until the end of the molecule. In an insertion, an extra base is inserted into the sequence. In this example, a T was inserted between the first A and C in the first codon in the DNA strand, which changed every single amino acid that was produced. So instead of the ones up top, every one below is now a new one. So that changed the entire strand. That protein is not going to do what it was originally intended to do anymore. It's going to be a completely different structure. In a deletion, the loss of a single base is deleted and the reading frame is shifted to the left. So here you have a sentence. The sentence is representing codons. Instead of using the A's, T's, C's, and G's that you have in DNA, it's using just letters from the alphabet to create words that you'll recognize to see how it changes the words. So the original sentence was the fat cat ate the rat, and if we delete the H and then shift everything over to the left, and we're still reading, every codon is 3 letters, so we just have to do the next 3 letters. Now you have teth, atke, ada, tet, her, at. It doesn't make any sense anymore. It's no longer a sentence. It's not going to do the same thing it originally did, or convey the same message, or make the same protein. So these are examples of substitution, deletion, and insertion with a sentence. The dog ate the fat cat. A substitution would change the sentence to the dog ate the fat rat. We just substituted out the C for an R. The sentence still makes sense. It only changed one word. So, maybe it still works. It's still a valid sentence. So we don't know whether or not that's going to have a really detrimental impact on the end product. Remember that many codons code for the same amino acid. So depending on the change, you may still have the same amino acid there, and the protein may end up being the same. The deletion, we deleted the D in dog, and so you get the sentence, the aga tet hef atter at. Well, that's not really a sentence anymore. It doesn't make any sense at all. It changed everything from the word dog on, and the protein is going to be completely different as a result. The insertion, we inserted a T before the word ate, and it changed the sentence to the dog tat eth effa tra with a leftover T. Again, the insertion shifted the reading frame to the right 1 letter, and as a result, it changed every codon beyond that point. Chromosomal mutations are different than point mutations in that they impact the entire chromosome. A chunk of a chromosome is changed, which could be hundreds, if not thousands, of nucleotides large. So the chromosomal mutations involve changes in the number or structure of chromosomes, and they include deletions, duplications, inversions, and translocations. This is a picture of a deletion. The original chromosome up top has A, B, C, D, E, F, and then the deleted, the chromosome that had a deletion in it on the bottom has A, C, D, E, F. So you see the section B was removed entirely, and the new chromosome is missing a chunk of information, and it is shorter as a result. So that's a deletion. A duplication produces an extra copy of parts of a chromosome. So here you have A, B, C, D, E, F as the original, and the duplication is A, B, B, C, D, E, F. So you have the B chunk was duplicated and put back in there an additional time. That can occur more than once, so you could have like BBB, or just like AB. You can have a larger chunk, but a duplication is any time that any section of a chromosome gets made again and put in there. So the end chromosome after the duplication is larger than the original. An inversion is when a piece of the chromosome is taken out and it's reversed the direction and put in backwards or upside down. So you have A, B, C, D, E, F here, and in the inversion you have A, E, D, C, B, F. So that whole B, C, D, E section came out, flipped around, and went back in as E, D, C, B, completely backwards. Or inverted. If you think of inversions that people do, a handstand would be an example of an inversion. So just going upside down, the complete opposite. You should notice that the original chromosome and the chromosome after the inversion are the same length. Whereas with the deletion, after the deletion it's shorter, and with the duplication, after the duplication, the next chromosome is larger. With an inversion, they're the same length because you're just taking out that one chunk and flipping it. So there's the same amount of information there. Now it's just in a different order. With a translocation, this involves two non-homologous chromosomes. So the original chromosome, you have A, B, C, D, E, F. And then with a translocation, a chunk of that chromosome can actually transfer to a different chromosome, or maybe trade places. So here you have DEF breaking off the first chromosome and being transplanted on the chromosome below, while that JKL came from the bottom chromosome and went on the top. So anytime you're thinking about a translocation, think that it actually transfers locations. So it is going from two non-homologous chromosomes, meaning they are not the same. So you could have a piece of information from chromosome number 4 going on to chromosome number 2, and the piece from number 2 going on to number 4. It's where two chromosomes exchange bits of information. That is a translocation. Many mutations have little or no effect on gene expression. So some cause genetic disorders. And polyploidy is a condition in which an organism has an extra set of chromosomes. Down syndrome is the most well-known example of polyploidy. It's called trisomy 21, where you have 3 copies of chromosome number 21. And we'll talk more in depth about different examples of polyploidy and what they may mean in a later lecture. Some mutations have little or no effect because some mutations don't result in a change to the end product or a change in the protein. Many of us, actually all of us, have some sort of mutation in our body, and many of them are not important or not detrimental at all. Whereas some genetic mutations, like the point mutation that causes the disease Tay-Sachs, or cystic fibrosis or anything, they can be very detrimental to your health and very dangerous. So mutations may be silent or neutral and cause no impact whatsoever. They can be harmful or fatal, or have a very bad impact, like in the instance of Tay-Sachs, or they could be beneficial, but there's a very low probability of a mutation being beneficial. We're not creating X-Men around here. So with this picture right here, take a moment. What would you call this type of chromosomal mutation? It is a chromosomal mutation. All right, and what would you call this chromosomal mutation? And this one? Remember, they're both the same length. Yep, that's an inversion. And last but not least, so A is your deletion, B would be your duplication, C is a copy of your inversion, and D is your translocation, the 4 different types of chromosomal mutations. And just to review, the 3 different types of point mutations are substitutions, insertions, and deletions. And the insertions or deletions do cause a frame shift mutation. Have a great day.